The cells within our brain transfer information via tiny communication hubs called synapses. At these synapses, a protein called Amyloid Precursor Protein, otherwise known as APP, sits within the membrane. Here we show part of full-length APP. The rest of the protein extends off screen. You can see the parts of the protein inside and outside of the cell. Talin is an essential scaffold protein which connects the cell's force-generating machinery to the synapse. We only show parts of talin that interact with APP, which you can see in green. Previously, we have shown that talin and APP interact with each other. In normal, healthy cells, we believe the talin-APP linkages are under tension and connect both sides of the synapse. We will now show the events involved in APP processing. APP is processed by a family of molecular scissors, otherwise known as enzymes, called secretases. The first processing step. Here we show what APP looks like after it's been cut by the beta secretase. The APP protein moves within the membrane due to forces coming from inside and outside of the cell. Here you can see forces from inside and outside of the cell pulling on the APP, moving it up and down in the membrane. At this point, the APP can be cut in two ways by the gamma secretase complex. The key byproducts of APP processing are amyloid beta 140 and amyloid beta 142. Amyloid beta 42 has been heavily implicated in Alzheimer's disease. Here we see the two cutting sites that lead to different types of amyloid beta being produced. These cuts are made by the gamma secretase, which is composed of nicastrin, presenilin, APH, and PEN2. The enzyme's active site is shown in red, and the two sites of which it can cut at are shown in pink and black. The pushing and pulling of APP means that the enzyme can cut at different sites. You can now see the second cutting event. Cutting at the pink sites initiates a processing pathway that produces harmful amyloid beta 42. Because of this cut, the part of APP inside of the cell moves away from the membrane. It's likely that talin is released at this point. Two further cutting events occur that ultimately lead to the production of amyloid beta 42, seen in pink and dark pink. In normal, healthy synapses, tension and mechanical forces are tightly regulated, maintaining balance across the synapse. We believe that when tension and forces across the synapse are uneven, APP is cut up in a non-normal and toxic way. This produces amyloid beta 42 that builds up into plaques, whilst also severing mechanical linkages leading to synaptic impairment. We believe that the talin-APP linkage and series of events shown in this video are central to the early cellular development of Alzheimer's disease.